Very happy afternoon, students. Welcome back to RGR Academy. So this is a biology session. I hope you're all safe at home, and we'll start with today's biology session. Before we'll start with a new chapter, that is plant growth and development. So before we start with this chapter, let us have a quick recap of what are things that we discussed in the previous class. We are in the plant physiology, the new unit of this particular um, chapters by we discussing, where we discussed about photosynthesis, the respiration, and we discussed about the previous chapter, that is we discussed about the biological, that is nitrogen metabolism that we discussed. And yesterday especially we discussed about the nitrogen metabolism. We discussed the nitrogen cycle, the nitrogen fixation, the root nodule formation, and we discussed stages involved in the nitrogen fixation, where ammonification, nitrification, denitrification, and nitrate assimilation, where we discussed like how the ammonia is getting converted to nitrite and how nitrate is getting converted to nitrate, and we discussed the fate of ammonia, and at last we discussed the heterotrophic mode of nutrition. So this is what we discussed, and we completed that particular chapter, that is mineral nutrition, as well as mineral nutrients that we discussed along with the nitrogen metabolism. So almost we're done with 3 chapters in this particular unit. We have other 2 more chapters. The other chapter that we're going to discuss today is plant growth and development. So coming to this particular chapter, that is plant growth and development. What we're going to discuss in this chapter is we discussed what are the factors which usually stabilize the growth of the plants, and what is exactly mean by growth, or the types of growth like logistic growth, exponential growth that we discussed, and we'll discuss about the measurement of the growth, how the relative and the absolute measurement that can be performed, that we'll discuss in this chapter. Also, we discussed about phytohormones, the growth regulating factors, that plant hormones like auxins, gibberellins, cytokinins, abscisic acid. These are all the different hormones that we're going to discuss in this chapter. And also the germination. Now coming to the germination part, we discussed about hypogeal germination and epigeal germination. And we'll see also about the seed dormancy. So seed dormancy, what is exactly the seed dormancy? And what are the significance of the seed dormancy that we'll see? And vernalization. We also discussed about vernalization. Coming to the vernalization, that how we exposing the plants with the cold, that is low temperature, right? So the significance of the vernalization that we'll discuss, plasticity, heterostyle that we'll discuss, and also we discuss about photoperiodism. Under photoperiodism, the short day plants, the long day plants, and neutral plants. These all things that we're going to discuss. So, on the whole, we try to complete this chapter by today. That is plant growth and development. If time permits, we try to complete. If not the case, we extend this chapter for the next session as well. So, as far as this chapter is concerned, that is plant growth and development is concerned. Plant growth and development. The questions from this part, you get like 2 to 3 questions that you can get from this chapter. One main question that you get from the hormone side, from either like an auxins, gibberellins. Definitely one question will come from the hormone side, its function, like which hormone induces like fruit formation, which hormone for senescence formation, which is for like aging of plants. So from the scientist like who discovered this particular phytohormone, from this area you can get a question. So definitely one question you get from the hormone side. The other part, when you're talking about the growth, so growth area that you can get a question. So maximum of 3 questions that you can get or expect from this particular chapter. So let us begin with this chapter, that is plant growth and development. So let's begin with this chapter, the plant growth and development. Plant growth and development. Okay, so we'll start with this chapter. So you know pretty well what is exactly the growth we're talking about. Growth is actually the irreversible increase in the size. So this is what actually growth is an inevitable thing as we could see in case of both plants as well as in animals. When both the cases you could see the growth is an essential thing. And as far as the plant growth is concerned, how can you define the growth? Growth is something it's a irreversible increase in size of an individual cell or organ or its part. So this is what actually that we can, it's an irreversible. Once the growth is attained, like which cannot be taken it back. So it's an irreversible increase in the mass of the cell or a tissue. So substantially what will happen, this keeps on increasing in size as well, and the development takes place further on. So when we talk about the characteristics of the growth, we talk about the characteristics of the growth. Coming to the characteristics, so first and foremost thing in the characteristic is, as far the growth is concerned, the growth is indeterminate. But in few cases where you could see the growth can be determinated. So as I told you, like growth is indeterminate, like you could see in case of meristem tissues where you could see it's a self-perpetuate. But where you could see in the plant structure, there is a leaf, flowers, and fruit. These leaves, flowers, and fruit, this has got determinate. So the leaves, flowers, and fruits, these are all determinate. But when you take the meristematic tissues, especially you take the meristematic, I'm talking about indeterminate. When you talk about like meristematic tissues, these tissues of the plant which seems to be indeterminate, but exceptions where you could see the leaves, the flowers and fruits, like again, these parts of the plants which seems to be determinated. And also you could see the plant structure, like the stem roots you take. Stem roots again, it's like indeterminate growth. So meristematic, and you got to see the parts of the stem where this seems to be indeterminate growth is achieved. So first characteristic of the growth is it is indeterminate, and second is growth is measurable. You can measure the growth. Growth is measurable. So how do you measure the growth? By means of cellular level growth, or increase in cell number. So the cellular level of the growth can be identified, and also you can measure the growth in terms of increase in cell number. Increase in cell number. Okay, increase in cell number. Today we have class till at 4 o'clock. We have till at 4. So cellular level and increase in cell number. So increase in cell number or increase in size. In this you will be calculating the dry weight and the fresh weight. And you'll be calculating the fresh weight of the plant. So that is what increase in cell number. Coming to cellular level growth, length, diameter, surface area, you'll calculate in terms of length, that is surface area. Length, the surface area which can be calculated. So these are the two main features that you could see as far as growth is concerned. One is growth is indeterminate, other one is growth is measurable. So either you take, as far as like cellular level is concerned, you go with the length or go with the diameter or go with the surface area. But increase in cell number, the dry weight you take the fresh weight, or you even take the volume of the cells. You take even the volume of cells. So this is just an introductory part of the growth and the development. So growth is concerned, the characteristics, we have many other features, but when I'm making very precise, the growth is indeterminate and growth is measurable. Okay, so now we move to the phases of growth. As far as the phases of growth is concerned, we have three phases. Can someone tell me what are the three phases of growth? So in this slide, you could see there are three phases of growth. So this we discuss about the phases of growth. Coming to the phases of growth, there are three phases. The cell differentiation region we have, the cell elongation region we have, and then we have the cell division. So cell division, cell elongation, and this one. So by coming to the elongation part, so what are the things that are going to take place in the elongation region? So coming to this part, cell elongation. So as far as cell elongation, what are the main things that is going to take place in cell elongation region? Usually the meristematic cells usually involved in this way. Cell elongation, where you could see meristematic. Here you could see the meristematic cells. And these elongation regions are highly vacuolated. The cells are highly vacuolated. I'm talking about the features of the cell elongation region. Okay. cell elongation region, we're talking about the plant growth and development. This topic is a plant growth and development. Okay, so usually a cell elongation region which consists of meristematic cell and it is highly vacuolated. This is highly vacuolated, and here the cell wall which starts accumulating. This here the cell wall which starts accumulating. These are the features that you could see. Starts accumulating, accumulating in this case, where in cell elongation region. Coming to the cell division region. So cell division that you could see like the maturation which undergo the structure that is functional differentiation, where you could see the functional differentiation is attained. Functional differentiation. In this place, you could see the functional differentiation can be attained, and the cells which goes to the special tissues, and where you could see no more growth afterwards. One particular time, like growth is reached. For example, you take xylem and phloem. You could see like maturation phase. Once the maturation is attained, so once the complete maturation is attained, after which what will happen? No more further maturation, no more further development that you could see. That is a maturation that once it attained, no more growth afterwards. For example, you take in case of xylem. You take in case of xylem and phloem. Xylem and phloem. Coming to this part, like cell differentiation part, like you could see like phases where we discussed about elongation, we discussed about the maturation phase, and cell differentiation. Here the cell will become specialized in this case. So on the whole, how do we categorize the phases of the growth? We can categorize the phases into three. So one is meristematic. We can categorize into meristematic, and second one is elongation, and third one is maturation. So these are the three important phases that you could see as far as the plant growth is concerned. So okay, fine. Here we have the meristematic, we have the elongation phase and the maturation phase. Usually the meristematic phase, where you could see in the shoot apex. You should see in the shoot and the root apex region. Meristematic, this type of growth, the phase that you could see usually in the shoot apex and the root apex, where there is a presence of what? The primary cell wall. In this, where you could see the presence of primary cell wall, and also in the meristematic rate, meristematic phase, the respiratory rate is more. So these three things, since this is like a clump, I'll repeat once again. The meristematic phase is concerned. The meristematic phase is, you could see the shoot and the root apex region, and the meristematic phase that you could see the presence of the primary cell wall, and the RR means respiratory rate. The respiratory rate which will be increased in the meristematic phase. Okay, and I told you already about the elongation, which is consists of meristematic cells, and you could see the highly vacuolated, and the cell walls start accumulating. And coming to the maturation phase which undergoes the structural and functional differentiation, which undergoes structural and functional differentiation. So once it attains the structure and functional differentiation, that once it attains a maturity, then after that, once it reaches specialized tissue, for example, you take xylem and phloem, after that what will happen? There will be no more growth afterwards which takes place as far as the maturation is concerned. So very shortly we're going to discuss this related to this, like differentiation, redifferentiation, dedifferentiation, all this part that we're going to discuss. In that part, you can clearly understand. Okay, so as of now, this slide represents the three phases of growth. What are the three phases of growth? The first one is meristematic phase, number two is elongation phase, and third one is the maturation phase. I already gave the points. Here the RR represents respiratory rate. In meristematic phase, you could see the respiratory rate is more, and there's the presence of the primary cell wall. And here you could see the shoot. You could see the shoot and the root apex region. Fine. So where you could see the vacuolated cells, you could see the cell elongated region. So from this, you get a question, maybe like which among the following is a correct statement or incorrect statement. In this type of question comes like you must be in a position to answer. All right, guys. So we're done with the phases of growth. Next, we move on to the types of growth. Coming to the types of growth, we have two types of growth. What are the two types of growth? We have arithmetic growth and the geometric growth. So coming to this, the growth is concerned. This slide shows the types of growth. The growth types. We have arithmetic growth, and we have a geometric growth. Fine. Can someone tell me like what makes the main difference between the arithmetic and geometric growth? if you start plotting the graph for arithmetic and the geometric growth. So what makes the difference? Like if you're plotting a graph, for example, if you plot a graph for the arithmetic growth, you get structure like this. But if you plot a graph for geometric, you get the lag phase, then the log phase, then the stationary phase, and sometimes it will go even go to the decline phase. So this is how we could see the difference between your arithmetic growth and the geometric growth. So here in this case, arithmetic, you could see the curve is, the graph is linear. Here the graph is linear. The graph is linear, but you could see like, S shape. You could see the S shape, a sigmoid shape curve that you could see in case of geometric. Okay. So I'll definitely send the pictures based on the request. If you just send a hi or something like that to my number, definitely I'll send all the pictures, because I should know to whom I need to send the pictures. Definitely I'll send the pictures. If you request or give a signal like, you need a pictures in WhatsApp, I'll send pictures for you. Okay, Priyanka? Fine. So arithmetic and the geometric. When you plot arithmetic, what happens, like, it will be increasing in an arithmetic manner, like an additional that you could see, like, so one cells which keeps on increasing, like, you could see 3, then 4, and then 5 and 6, one numbers which keeps on increasing in a arithmetic form. But when you plot the graph, what happens, like, if you could get the linear curve, if you could get the linear graph that you could see in case of arithmetic. But in geometric, practically speaking, you could see this phase, you could see lag phase, lag phase. This is logarithmic phase, and this one is a stationary phase. So this is quite common phase that you could see the geometric, where if any organisms you take in a bacterial species, if you just try to inoculating in a particular petri dish or something, immediately the bacterial will not start growing. It will go with the initial phase, we call it as lag phase. So there will be a lag phase, and then there will be increase. There is increase in number, where this phase is called as logarithmic phase. Usually, logarithmic phase, what type of metabolites usually produced? Primary metabolites. Primary metabolites usually get produced where the cells, the numbers start increasing due to the production of primary metabolites. Once it reaches the stationary phase, then the organisms start producing the secondary metabolites. They start producing the secondary metabolites. Sometimes the secondary metabolites even seems to be toxins. Sometimes seems to be toxins. Once the cell reach the stationary phase, there won't be increasing in cell mass, there won't be increasing in cell number. So in geometric, you could see this type of sigmoid curve, lag phase, log phase, then it goes to the stationary phase. One point of time, the situation seems to be like unfavorable. Okay, unfavorable. Then what happens, like, the phase which the organism, the number which drops down, then it goes even to the decline phase, right? But when the thing which goes to the linear graph that it goes, like, for example, you go with the you go with the fermentation, like fermenter process, that bioreactors techniques, where you'll be supplying the nutrients continuously, right, until you reach the products. You'll be supplying the nutrients continuously. You keep on making the organisms to multiply. In that point of time, what happens, like, automatically the cells which keeps on increasing in size usually get only the linear. In case you go with the reactors, bioreactors, in all these cases, if you go with this, you can see the linear, where the organisms which will not go to the lag and log and the stationary in the artificial environment, where you could see in case of fermenters and bioreactors that you cannot see this. But in normal situations, the practical speaking, you could see the lag phase, then log phase, then the stationary phase. If the organisms is like, there's shortage of like nutrients or unfavorable conditions, again, this even go to the decline phase. And logarithmic phase, where the primary metabolites can be produced, on this stationary phase, the secondary metabolites can be produced. So as for the growth types are concerned, we have two types. One is arithmetic, other one is the geometric type. So where you could see the arithmetic, the linear type of the graph that you could see, where you could see in a root elongation. You actually could see this type of growth in a root elongation. You could see this arithmetic type of growth. Coming to the geometric, you could see mostly in the cell tissues and the organs. You could see in the cell tissues, and you could see in case of organs. So when coming to the difference, the increased growth per unit time is called the growth rate, that actually. This growth rate can be arithmetic or geometrical. That's what we are discussing so far. And arithmetic growth phase is only one daughter cells continues to divide. So here only one daughter cells is continuing to divide, and other daughter cells get differentiates and matures. I told you already, this is for elongation of root, where you could see the arithmetic growth that you could see in the elongation of root. Coming to the geometric growth, I told you, like, we have the phases like lag phase, and we have the log or exponential phase, and we have the stationary phase. Examples: the cells, tissues, and organs. This comes under the geometric. But when you plot a graph for arithmetic growth, you get the linear type. But when you plot a graph for geometric, you get the sigmoid, like, lag phase, log phase, and a stationary. So this type of their shape like you get in case of geometric curve. So where you could see the arithmetic elongation of root elongation. Geometric growth, where you could see in the cells, tissues, and in organs. So these are the two types of growth that you could expect. We have the specific formula also. Can someone tell me what is the formula that you usually we plot for the arithmetic growth? As far as arithmetic growth, LT is equal. Can someone tell me like what is the formula for arithmetic? LT is equal to LO. The formula for arithmetic. The formula is what? Usually the LT is equal to LO plus RT. LO plus RT. So this is the formula that usually they plot for arithmetic. Coming to the geometric, what is the formula? W1 is equal to WOERT. W1 is equal to WOERT. So this is the equation that you can give for the geometric. Okay. So for geometric, W1 is equal to WOERT. So the expansion of what is LT, LO, that RT, that natural algorithm, everything is given in the textbook. So and let me not waste my time like giving all the expansions again. It's a time consuming. For that, I'm just giving only the equation for arithmetic and for the geometric. For geometric, if they ask for the equation, it's like W1 is equal to WOERT. Okay. So coming to the arithmetic, it is LT. LT is equal to like LO plus RT. Okay, this is what the equation that you should remember. Okay, so if you want to know what is W1 and WOERT, what is like LT, RT, then everything, like you can refer a textbook because it's already given both state board as well as in the NCERT books. Okay, so we're done with the types of growth, arithmetic and the geometric growth. Now, next we move on to the rate of growth. So how the rate of growth can be accomplished? How do we calculate the rate of growth? So coming to this part, the rate of growth. We have two types of the rate of growth can be measured. What are the two types that you could see? One is absolute growth rate, other one is relative growth rate. One is absolute growth rate, and the other one is relative growth rate. So questions sometimes which comes in the calculative part. Okay, so how do you calculate the absolute and how do you calculate the relative growth rate? Absolute growth rate is what actually the total growth takes place. It's a total growth on the whole. So the total growth. But when you come to the relative growth, so when you talk about when you talk about the rate of growth, what we should remember is increase in the length or weight of the plant part per unit time. That is actually the definition of the rate of growth. What is how the rate of growth can be defined? It is increase in the length or weight of a plant part per unit time. This is what actually the definition says. Based on which this can be differentiated into absolute growth rate and we call it as relative growth rate. We call it as RGR. RGR is relative growth rate. Absolute is AGR. Absolute growth rate is we call it as AGR and RGR. Fine. Coming to this part, when the calculative part comes, like how do you calculate? If they give this type of diagrams, like they ask you to identify the absolute growth rate and they ask you to calculate the relative growth rate. So absolute growth rate calculation is very simple. So how do you calculate? For example, this diagram they given. This is the initial part where you could see the green part. If they ask you to calculate the absolute growth rate. So how do you calculate that calculation, which is based on what? 10 centimeters square. 10 centimeters square. That is minus 5. 10 centimeters square minus 5 is equal to what? Minus 5 centimeters square. This gives 5. So this is 5 centimeter square. If you make the difference between this 5 centimeter square and the 10 centimeter square, this gives a difference. This is how you normally calculate the absolute growth rate. Okay. If you make the difference, it's very simple. When you calculate the absolute growth rate. For this example, you take, if you want to calculate the difference, then how do you do the 55 centimeter square minus 50 centimeter square? Okay, this gives what? This gives 5 centimeter square by t in a given period of time t. For example, you take 7 days, right? The given period of time is t. So 5 centimeter square by t. This is the absolute growth rate. The same way here you could see like 5 centimeter square divided by t. So this is the absolute. If they ask you to calculate the relative growth rate, then what you have to do is we need to calculate. So you get the 5 centimeter, right? So you need to calculate this 5 centimeter, for example, in this case, here you could see the 5 centimeter. This is what absolute growth rate. 5 divided by this initial growth. 5 divided by 50 into 100. This gives what the answer that you get, right? That is your relative growth rate, RGR. So it's something, the difference, like the initial point of time, what the growth you got, that is 50, right? So, and what is the difference that you get in the absolute growth rate? This is absolute growth rate and the initial growth you got, right? That is 50. So you just divide. So what happens, like this will get 50 and this will get 2. So 5 into 2, this equal to 10 for this case. This in case you could say 10. This is your relative growth rate. In this example, if you see, this is how you calculate. Here is also 5, here is also 5. So what you get is like 5 divided by 5. So 5 divided by 5 into 100 also, if you make, if you don't want to calculate the percentage, then 5 into 5. Then it's actually like 551. So this will give a 100%. Getting? So this is your relative growth rate. So questions mainly comes from relative growth rate, because calculating absolute growth rate is very simple, because you're just going to make the difference, like in 10 minus 5, that is like your absolute growth rate. That is very easy. But when the question comes to calculate the relative growth rate, first you should calculate the absolute growth rate. So you got the absolute growth rate, and then what is the initial growth has been achieved? It is 50. So you make the difference, like 5 by, that is the difference, like 5 by 50 into 100. So if this comes, because already you calculated the absolute growth rate is 5, that is divided by this one, the inner surface, the initial growth, right? The 50 into 100. You cancel it and multiply 5 into 2. Here, if you calculate here, the percentage is 10. But in this case, you could see the percentage is 100. 100 is relative growth rate. In this case, in this example, the relative growth is 10%. So it's a very easy calculation, but you need to know how you should calculate the absolute growth rate, that is, and how you to calculate the relative growth rate. Usually the questions may not come from the absolute because it's very easy. But when they're asking for relative growth rate, first you should calculate the absolute growth rate, and having that value, and then going with the initial growth, then you just divide it by that value and then make it as 100. If you make it as 100, you get the percentage. So here RGR is 10%. Here the RGR is 100%. So it's a very easy calculation by which the rate of growth can be determined. Okay, so this slide which represented the difference between the absolute growth rate and the relative growth rate. What is the absolute growth rate? Just making the difference between these two, right? Just minus, okay, 10 minus 5, that's all. That is the absolute growth rate. Having that as a value, that value is just divided by this one, the initial growth, right? Then you get into multiply into 100. So what's the percentage that you get? That is the relative growth rate. In this case, the relative growth is full 100%. In this case, the relative growth rate is only 10%, because here you took 50, here you take 5. Okay, you divided in this case by 50, here you divided by means of 5. So this made the difference between the absolute growth rate and the relative growth rate. In terms of formula for relative growth rate, growth per unit time, I just give the formula for RGR. Okay, if you need a formula for RGR, RGR is growth per unit time. Growth per unit time. Growth per unit time, which is divided by, growth per unit time is divided by initial parameter. Initial parameter. What is the initial parameter that we're talking about? This one, right? That 50 or this 5. This is the initial parameter. Growth per unit time, that's the difference that you got before, like the difference that you got, and this is absolute growth rate, the difference. And the initial parameter, it may be a 5 or a 50. For example, in this case, how you calculated? You calculated that is 5 divided by 50 into 100. So this is given what? This is given 10%. This is given a 10%. That this is your relative growth rate. Okay, so RGR is used for relative growth rate. Which part? Relative growth means you make up the difference. You make up the difference relatively. You need to make up the difference, but absolute is just making the difference between, like, this part and this part. 10 minus 5, that is an absolute growth rate. But relative is actually with the difference. For example, 10 minus 5 is 5, and this 5 has to be divided by the initial parameter. The initial parameter. So then if you give, in this case, like, this gives 1. If you multiply into 100, this gives the 100%. So that also, like, if they're asking in terms of percentage, if you need to multiply into 100. If they didn't give the asking for percentage, just Score of this one, you get the value just 1. Okay, okay, only in leaf. So this usually be done mostly in the leaves actually. Usually in case of leaves that you can perform the relative growth rate. It's not we cannot conclude only we can do it only with the leaf. We cannot limit ourselves like various phases where you could see in case of stem, the other group of cells also we can calculate, but based on the dry weight and the flesh weight, using this we can calculate. Okay, the parameter is usually is the time. Usually you go for the time, either you take the 7 days of time or you take the time, the factor. Usually the time factor is actually the time, the parameter is the time. Okay, so this is how you can give. For example, in this case, the time is given as 7 days. The time, the parameter is given for a 7 days. Then you need to determine whether you're giving the parameter the time is like 7 days or given is like 14 days. That you can determine based on your study. Fine. So this is just a calculative part. I hope you guys can understood now how to calculate the absolute growth rate, that is AGR, and how to calculate the RGR. So to calculate RGR, you need first AGR. First you need to calculate AGR, and then you can calculate the relative growth rate. Fine. We're done with this part. That is the rate of the growth. Okay, so I think you asked for the formula. So once some of you asked the formula, I told you already. Coming to the formula, arithmetic, the formula is LT is equal to LO plus RT. Coming to geometric growth, it's W1 is equal to WO ERT. So this I've shown you already. Okay, fine. So guys, we move to the next part. So the rate of growth that we have completed. Next, conditions for growth. What are the conditions which is required for the growth? You might be knowing what are the conditions which are required for the growth. The conditions which are required for the growth. What are the conditions? You take the light is a main important factor. The light, then we have a temperature. Now you're going to tell me what are the optimum light, what is optimum temperature that you're going to tell me. So light, temperature, you take water. Water is one more important factor. And then oxygen, then mineral nutrition. So we discussed the last chapter, mineral nutrition, whether the macronutrients or the micronutrients. So mineral nutrition, then growth factors, the growth factors, and genetic factors. These are the factors which are responsible for the plant growth and development. Okay, so what are the factors, guys? Light, temperature, water, oxygen. You take mineral nutrition. GF is the growth factors, that is the plant growth factors, the hormones, and we have the genetic factors. Coming to the light, the proper optimum light is required. Fine. So coming to temperature, temperature is concerned 20 to 35 degrees is actually the optimum temperature, 20 to 35 degree centigrade. If it goes beyond this, if it goes to 45 degrees, then what happens? This will damage the protoplasm of the plant. If the temperature, if it goes beyond the 45 degrees C, then what happens then? It damages the protoplasm, and also there are chances of the proteins, also the enzymes can be denatured. So temperature is 20 to 35, but above 45 it goes where the protoplasm function can be malnourished. Then coming to the water part, water is very important for turgidity, to maintain the turgidity of the cells. Turgidity. Okay. Then we discussed about the oxygen. Oxygen is for the metabolic energy. For all the metabolic energy, it requires oxygen. So energy, metabolic energy, I'm dictating because it's not clear. Metabolic energy. For this, it requires oxygen. Then mineral nutrition, you studied already, all the macronutrients, the micronutrients we discussed, especially you take sodium, potassium, copper. These are some of the nutrients which are required for the growth and development of the cells. Then the growth factors that we're going to discuss in detail, the plant growth, or PGF we call it as plant growth factors, or the hormones we discuss like auxins, gibberellins, all the things that we're going to discuss very shortly. And the genetic factors, these genetic factors are nothing but the intrinsic factor. All the intrinsic factor, this is a combination of both intrinsic and extrinsic factors that we are talking about. So genetic factors, something like intrinsic factors, something which is present within the cell. This also plays a very important factor in determining the favorable conditions as for the plant is concerned. Okay, clear guys? So this is about the conditions for the plant growth. Then how do you go for a measurement? So this is one type of measurement that we have for the leaf is concerned. So what type of measurement? That is AGR and RGR. This we did. When coming for the general about how do we go for the measurement? The measurement is concerned. I'll just tell you what are the different techniques by which you can go for measurements. Fine. So coming to another measurement area, since I don't have place, I write it here. measurement. So what are the different techniques by which you can go for a measurement? So how do we measure? By means of increasing length, surface area, or a volume. By means if it is increasing length, or by increasing surface area, or by increasing volume, by increasing volume. By these means we can go ahead with the measurement. By means, how do we do? We by means of dry weight and by means of diameter, increasing in diameter. Then you go with the fresh weight, then and the fresh weight of the plant as well. Using what is the device that instrument that you use? Oxonometer. Oxonometer. Usually by using the oxonometer, we can go with this like oxonometer. Then we can use horizontal microscope. We have the horizontal microscopes. Microscope. This can also be used. Also, you can use the crystallograph. We have crystallograph. In this like crystallograph way, the magnification is up to 10,000. The magnification is more than 10,000. So 10,000, the magnification is more. So these are the different. Nowadays, we also could see like the digital techniques. It's also coming. It's like very old techniques that, of course, I can understand, right? And there are many digital experiments by using the probes also nowadays. Like for example, if you go with the bacteria, so you want to calculate the bacteria has been grown like numerous in the petri dish. We cannot count one by one, two, three, four. That we cannot count, but that like half a day will go, right? There is like a technique where we have a probe techniques, like we have digital pen systems, like just pressing in that particular area, the digital techniques, the probe detecting techniques, we can able to even identify. There are like many modern techniques also quite available, right? Okay. So there are modern techniques also available by which we can able to identify apart from the dry weight, the diameter, the fresh weight, using oxonometer, horizontal microscope, the crystallograph technique, beyond which we also go with the other techniques by which we can calculate or we can measure the growth of the plant. I hope, Christian, my voice is audible for you. Okay, right. If it is like feeble, you tell me so that I can increase my voice. Okay, thank you. So by means of advanced technique also we can do it. Fine. This is about the measurement. Next, coming to the differentiation. So coming to the differentiation part is concerned. So we move to the next part, the differentiation. So let me erase this part. So we're done with this. So here, development. As far as development is concerned, coming to the development, we have three types of differentiation, de-differentiation, redifferentiation. So first one is differentiation. You sometimes you may get confused. What is differentiation? And we use we use the other technique like de-differentiation. Other process we call it as de-differentiation, and redifferentiation. So let us discuss about these three: the development, differentiation, de-differentiation, and a redifferentiation. So what is the differentiation, the mechanisms? So for this, you need a little bit of like understanding about the cell cycle. For example, you're talking about a cell cycle, the chapter, the cell cycle we discussed. So what are the phases? This is, you could see, this is the M phase. This is the M phase. And what is this phase? We call it as this preparatory phase. This we already discussed, guys. So tell me, what is this phase? This is the M phase. You have the prophase, metaphase, anaphase, telophase, and everything. This is the M phase, and this consists of, you have a G1 phase, and we have a S phase, and this S phase, and this is a G phase, and this one is your G1 phase, and this is your S2 phase. Okay, right. No, I'm not talking about a plasticity, guys. I'm not talking about plasticity. I didn't start with a plasticity. Okay, so next part we'll discuss about plasticity. This slide is about the development. Okay, about the development. So this is the preparatory phase here, where the cell is going to get, where the preparation is going to take place, right? This we call it as an interphase. I'm talking about normal cell cycle that we already discussed in a separate chapter. So interphase, now the cell is coming, and this is where the cell is going to divide. Here the cell is about to divide. So this is one cell. Okay, I'm talking about this cell, and this cell is going to divide. Okay, at the end of this phase, the cell is divide. So what is happening in this case, like differentiation, de-differentiation, and redifferentiation? the cells, it's already differentiated now. The cells, once it's going to, after division, like, now the cells have been differentiated. This process is quite common. This we call it as differentiation. So guys, you tell me if red is not visible. Okay, just to make the difference, I'm using the red color here. Okay, fine. Okay, so differentiation, just where the cell has been divided, where it's divided into two, this is the cells got differentiated. Then what, as the normal cells are divided, is called differentiation. So what is a de-differentiation is, then de-differentiation is once the cells actually divided, like, this may not be going back to the interphase again. What will be doing is, it will be going to the phase that we call it as a GO phase. I hope you can recollect. Okay, we can recollect that what is a GO phase. So instead of going back to the interface again, so what happens? This will go to the GO phase. So this particular cells which will be moving to the GO phase. So this process where in de-differentiation, what is going to happen? The capable of dividing cells regain their capacity of division under the certain conditions where this will not be going for a further differentiation. This will not be going for any more division. This will be going, it will be become a specialized structure. Once it be differentiated, this will not come back and it will not be differentiated again. So it will be going to the next phase that we call it as a de-differentiation phase. So what for that we need to make out the difference? What is the differentiation and the de-differentiation? Differentiation. So where this process occurs, differentiation, usually the normal cells. In the normal cells, you could see first I'll explain the differentiation. The normal cells which go with the differentiation with the regular, the cycle, with the regular cycle which goes with the normal cells. And this normal cells, what happens? This will be like, change to the trachyry elements. This change to the trachyry elements. It changed to a trachyry elements, and then it will become the elastic lignocellulosic material. It become a elastic cellulosic material. This is how actually the normal cell has got differentiated. Okay, normal, the difference, the cells got differentiated. But what is a de-differentiation? De-differentiation that you could see in this case, where the cells are capable of dividing, regain the capacity of division under the certain conditions. It means it will not go back to the interface again. Instead of going back to the interface, this cells, like, some cells which will be going to the GO phase, and there that will become a specialized cells. Understand, guys? Here differentiation, the cells have got differentiated. Fine. Cells have got differentiated. Okay, fine. Okay, sure, we'll discuss about it. So once it goes to the the de-differentiation phase, what happens? This will become a specialized structure. For example, I'm talking about the specialized structure. Here, for example, you're talking about the vascular tissues, you're talking about the bundles, you're talking about. This consists of your xylem, and we have got the phloem. Here got the phloem. Here we have the phloem, and we have the xylem. Okay, here we have the cambium. So fine. So now what happens? The normal cells, now it has become a specialized cells. Now it's not actually like, going to the interface. And for example, this, you take an example, I'm giving parenchyma cells. This is going to the parenchyma cells. Okay. So for the example, I'm talking like this is a parenchyma cells, where this is going to the next phase called as de-differentiation. De-differentiation. This is going to the de-differentiation. For example, you take a parenchyma. So the cells which is not going to it's not going to cell cycle, which is not getting divided, it's converting to the specialized cells. Actually, for the normal cells, now it has been converted to the parenchymatous cells. For example, you could see the interfascicular cambium, core cambium, all these things. So fascicular cambium, core cambium. So all the specialized structures are getting formed. These are the specialized structures which are getting formed after the differentiation. So this process is called as de-differentiation. So this is a de-differentiation. Which food will help for growth in our body? So you're talking about the minerals, you're talking about which food? For growth of a body, you're talking about the carbohydrates. In that angle, you're asking carbohydrates, proteins. The growth of a body, like, the carbohydrates and proteins. Yeah. Carbohydrates and proteins. The growth is mainly the proteins. Yes, fine. So this will become a specialized structure, that is a parenchyma cells. Then, for example, what is the main special structures which form cambium, core cambium, which is formed from the parenchyma cells. So this process is called as de-differentiation. So the cells got differentiated from that. So, but where it's going and doing its process, it's in the GO phase. It's into the GO phase, and the normal cells has become a specialized cells. For example, it become the parenchyma. Then what is the redifferentiation? Now what happened? De-differentiated cells again lose their ability. So again lose their ability. The cells again lose their ability. Which cells I'm talking about? Their ability. For example, you're talking about the parenchyma cells. This is what actually we discussed in the specialized cells. Again, the parenchyma cells which lose their ability, and this started forming the meristematic tissues. Now you see this started forming the meristematic, the parenchyma cells which started forming the meristematic tissues. By means, it's again started losing their ability to form the special. Again, it started losing this, the specialized structure. So this is called as redifferentiation. So concept is very simple. The concept is very simple. The normal differentiation, how the cell is dividing, is called differentiation. Once the differentiated cells which become a specialized cells, for example, which is going to GO phase and it's forming the parenchyma cells, for example, to form the cambium or core cambium, differentiation. Once the de-differentiated cells started losing their ability, and again, this started forming the other type of cells, for example, meristematic tissues. This we call it as a